Good morning, folks. Okay. Welcome back, everyone. How you doing today? Everybody's so quiet. It's a Monday morning, huh?
Good morning.
Good morning.
Good morning.
Good morning.
Good morning.
Welcome back, everybody. As we just wait a few moments to get other folks logged in here today, let me just say welcome back. Hope you had a good weekend. Weather was just beautiful out. I hope you had some time outside. Can you believe that we are starting our 5th week of the course, right? We are entering our 5th week of the course. Time really moves quickly, doesn't it? So, okay, we have an important week of activities ahead of us, right? This Wednesday is your first laboratory exam, right? This Wednesday is the first lab exam. And I'll keep reminding you, I'll remind you again on Wednesday, but that means on Wednesday at 9:30, you log into classmarker.com and take your lab exam, okay? And that's the only lab activity we're doing on Wednesday. So once you take your lab exam Wednesday, you have the rest of the time off. We're not meeting afterwards, okay? All right. Is there any questions about what's going to happen for the lab exam on Wednesday? Okay. Let me also remind you, we are now 2 weeks away from your very first lecture exam, right? Things are going to start accelerating. Things are going to start coming together, right? Assignments, exams are going to start happening. So 2 weeks to your first lecture exam, and you can expect that today, later today, I will place the study guide for the first lecture exam in the Files tab of your Lecture Wiley Plus course. Okay, so look for your study guide for lecture exam 1 by the end of today to be in Wiley Plus. Okay, do you have any questions right now?
I don't have a question, but I did want to say something.
Okay.
So I was going to get into like Wiley and then D2L and all that, and it said that my hack account had expired, so I had to reset my password, and it said that that's going to take 2 to 3 business days. So I don't know if that'll have any effect, like obviously not for the class marker, but I just want to let you know just in case.
Okay, Courtney, I don't anticipate an impact on what you're responsible for to me, but if you continue to have trouble with hack in some way, please let me know.
Okay, and then also for the lab exercise, it keeps saying that it's not available until the 29th of September.
Yes,
So
that was intentional. That's right, because I want you focusing on your lab test.
Okay.
All right. Any other questions? Okay, gang. Well, we're going to get into it here. We're going to finish chapter 3 today. The final major section of time that I'm going to spend with you is on the topic of protein synthesis, which in your textbook is around page 93, and it's going to cover several important figures: figure 3.26, figure 3.27, figure 3.28, figure 3.29, right? Lots of important figures here. So what I'm going to do today is there's some really big ideas here that need to be communicated. And so I think it will be helpful if you at least have seen a video, an animation, a cartoon that tries to share some of these ideas with you, and then we will talk about it extensively for the remainder of our time together. So I'm going to start today by showing you a video. Okay. And before I do that, I want to point out that in the class notes that I'm giving you, I've already placed the links for 3 different videos. So you will have them in your class notes and you can reference them during your private study time. The one that I'm going to show you at this time is the protein synthesis video. It's about 6.5 to 7 minutes long, but it's very important that you see this so that you can begin to think and understand about these topics. So let's, with no further ado, let's get that rolling here. Okay, folks, so here it is, DNA transcription and translation. This is the title of our first video. Here we go.
In order for our bodies to function, we need to supply them with a variety of nutrients we get from our diet. Our bodies cannot use the food as it is when it enters our digestive system. The process of chemical digestion uses different proteins and enzymes to break down the food particles into usable nutrients our cells can absorb. And where are the instructions to manufacture these and all the different types of proteins we need to stay alive? The instructions to make proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. For some genes, the encoded RNA is used to synthesize a protein in a process called gene expression. For these genes, expression can be divided into 2 processes, transcription and translation. In eukaryotic cells, transcription occurs in the nucleus, where DNA is used as a template to make messenger RNA. Then in translation, which occurs in the cytoplasm of the cell, the information contained in the messenger RNA is used to make a polypeptide. During transcription, the DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in 3 stages: initiation, elongation, and termination. During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. In order for the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as a 5' cap and a 3' poly A tail are added. This process is called intron splicing and is performed by a complex made up of proteins and RNA called a spliceosome. This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus through a nuclear pore and enter the cytoplasm to begin translation. How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3 letter codes called codons. The genetic code includes 64 codons. Most codons code for specific amino acids. there are 4 special codons, 1 that codes for start and 3 that code for stop. Translation begins with the messenger RNA strand binding to the small ribosomal subunit upstream of the start codon. Each amino acid is brought to the ribosome by a specific transfer RNA molecule. The type of amino acid is determined by the anticodon sequence of the transfer RNA. Complementary base pairing occurs between the codon of the messenger RNA and the anticodon of the transfer RNA. After the initiator transfer RNA molecule binds to the start codon, the large ribosomal subunit binds to form the translation complex, and initiation is complete. In the large ribosomal subunit, there are 3 distinct regions called the E, P, and A sites. During elongation, individual amino acids are brought to the messenger RNA strand by a transfer RNA molecule through complementary base pairing of the codons and anticodons. Each anticodon of a transfer RNA molecule corresponds to a particular amino acid. A charged transfer RNA molecule binds to the A site, and a peptide bond forms between its amino acid and the one attached to the transfer RNA molecule at the P site. The complex slides down 1 codon to the right, where the now uncharged transfer RNA molecule exits from the E site, and the A site is open to accept the next transfer RNA molecule. Elongation will continue until a stop codon is reached. A release factor binds to the A site at a stop codon, and the polypeptide is released from the transfer RNA in the P site. The entire complex dissociates and can reassemble to begin the process again at initiation. The purpose of translation is to produce polypeptides quickly and accurately. After dissociation, the polypeptide may need to be modified before it is ready to function. Modifications take place in different organelles for different proteins. In order for a digestive enzyme to be secreted into the stomach or intestines, the polypeptide is translated into the endoplasmic reticulum, modified as it passes through the Golgi, then secreted using a vesicle through the plasma membrane of the cell into the lumen of the digestive tract. Proteins are needed for most physiological functions of the body to occur properly, such as breaking down food particles in digestion, and the processes of transcription and translation make the production of proteins possible.
Okay, folks. So, quite a lot of unusual detail, isn't there? If you've never been exposed to this kind of content before in any other biology courses, this can be a little bit intimidating, okay? So what we're going to do today is make it less scary, okay? That's our goal today. Do you have any questions from having watched the video one time? Obviously, we're going to cover things, but is there anything you'd like to ask? Okay, so I'm just getting organized to take notes. Okay, so we're going to begin a lecture here on the topic of protein synthesis. Okay, that's our topic today. So let me say to you, first off, okay, the very first video referenced above is what we just saw, and it's explaining to you how proteins like insulin are manufactured by the cell's instructions, okay? Remember at the end of last class, we were talking about transport processes, and we did a little drawing where we had glucose in the bloodstream, and then we had insulin getting made in the pancreatic cell and it leaving the pancreatic cell. Well, at that time in that sketch that we did, we simply said, well, the pancreatic cell will make insulin, but we didn't describe how the pancreatic cell makes insulin. Today, we're going to describe how that actually takes place. Okay. Proteins like insulin, are manufactured according to the process described in video number 1 above. Okay. All right, number 2. The process of protein synthesis really is carried out by something called gene expression, right? Gene expression is the actual process that allows the cell to manufacture a protein, right? If you are on page 93 of your digital textbook at the subheading of protein synthesis, look for the bold-faced expression in your notes called gene expression. The process of gene expression is carried out by cells to make proteins. Okay. Okay, the process of gene expression takes an orderly sequence of events, kind of like it's kind of like an assembly line, right? If you've ever worked at a manufacturing plant of any kind, and you were a member of an assembly line, right? One person does something, and the next person does something, and the next person does something, and the next person does something. It's that kind of process that is used to actually manufacture a protein. Okay. Now, the order of steps that is taken to manufacture the protein is actually called the central dogma of biology. Okay. Central dogma of biology is the pathway of steps performed during gene expression to make a protein. Okay, so we have gene expression is something that you should be aware of. We have the central dogma is something that you should be aware of. And I'm going to give you 2 different versions of the central dogma. I'm going to give you the simple version of the central dogma first. Okay, the simple version is this. DNA is used by a cell to manufacture RNA. RNA is then used by the cell to manufacture protein. Okay, that's the simple version. DNA is used to make RNA. RNA is then used to make protein. Okay, that's a good way to start thinking about it. All right. Now, because of the detail that your textbook goes in, I'm going to go into even more detail than this. So I'm going to give you a detailed version of the central dogma. Okay, and to do that, it's just easiest if I do a little bit of drawing. So I'm going to pull my graphics tablet up here, and we're going to do a little drawing. Okay. All right. So here we go. Okay. So for the detailed version of the central dogma, once again, we're going to start with DNA. Okay. And we have to appreciate when we're talking about manufacturing a protein, when we're talking about expressing the information of a gene for the purposes of assembling a protein, okay, the DNA that we're talking about is actually a gene. Okay, so the DNA is, DNA is a molecule, and if I have a certain stretch of the molecule made of a specific sequence of nucleotides, which you remember from chapter 2, then you have a gene. The gene is the specific stretch of DNA nucleotides that tells the cell how to make the protein. So the process continues like this. The DNA of the gene is used to make immature RNA. Immature RNA. Well, yes, I'm just going to clarify that. Okay. It is immature RNA, and I'm going to say something a bit more precise. It is actually immature messenger RNA. Okay. The immature messenger RNA then goes through a process that we will describe where it is processed and it is linked back together in specific ways to develop mature messenger RNA. Okay. That messenger RNA is then exported out of the nucleus, and ribosomes will take that mature messenger RNA and translate it into polypeptide. Okay, the polypeptide will then fold into a very specific three-dimensional shape and become an actual protein. That protein will experience some additional modifications and then become physiologically active. Okay, so you can see that the detailed version of the central dogma pathway is quite a bit more involved than the simplistic version. So start with the simple version, get the basic idea, and then work your way out to understanding the more complete version. Now, let's stay with the detailed version of the central dogma pathway at this moment and add some detail. Okay. All right, so let's start at the beginning again. We take the DNA, a specific sequence of DNA we call a gene, and we use an enzyme to manufacture the immature messenger RNA. The process of taking the DNA and making RNA from it is given a name. It's called transcription. Transcription is the process of taking DNA and making RNA from it. So now I'm going to ask you folks, just because I need to know you're following along, okay? I'm going to ask you to take a look at figure 3.27. Take a look at figure 3.27. And here's a question for you in the chat window. Here we go. What is the name of the enzyme that makes the RNA? Can you tell me that from figure 3.27? What is the name of the enzyme that makes RNA? Figure 3.27, folks. What's the name of the enzyme that makes the RNA? Noah, you are not incorrect, but that's not what's written on figure 3.27. Okay. Come on, folks. All right. All right. Okay, so Karen and Kelly, what's the word in front of the word polymerase? Okay, thank you. There we go. That's what I'm fishing for. The answer is this. Okay, the name of the machine that makes the RNA is called RNA polymerase, right? The enzyme that makes the RNA from the DNA is RNA polymerase. Here's another question in the chat window. Here's another question in the chat window. How do I know that RNA polymerase is an enzyme? How do I know that? Okay, thank you, Noah. All right, everybody take a look at Noah's answer, right? That word polymerase, the last three letters of the word are ASE, right? And if you see a word with an ASE ending, you know it is an enzyme, because science has adopted the convention of naming enzymes with an ASE ending, right? So if you see polymerase, that's an enzyme. If you see isomerase, that's an enzyme. If you see kinase, that's an enzyme. You get the idea, okay? So one last thing in the chat window here, okay? What are enzymes famous for doing? What are they famous for doing? Now, why do we make a big deal out of enzymes? Okay, so thank you, Shay. Shay says that they synthesize protein. They are involved in that. And more broadly, what I'm trying to get out of you is that they increase the rate of chemical reactions. They accelerate chemistry, right? Enzymes are biological catalysts, right? Kendall, I see your comment, catalyze, right? Enzymes are biological catalysts. They make chemistry happen faster, all right? So what we're saying is, if RNA polymerase was not available to manufacture the RNA from the DNA, it could take a significantly longer period of time for the RNA to get made. All right, let's continue. Do you have any questions? All right, so we start with the DNA. We transcribe it using the enzyme RNA polymerase to make the immature messenger RNA. Okay, Kelly has a statement in the chat window. They're, meaning enzymes, they're involved in fermentation too, right? Yes, Kelly, very much so they are. All right, so the immature messenger RNA then has to go through a process known as RNA processing. RNA processing. Okay, RNA processing, your book does not have a separate figure for this. It does not have a separate figure for this. RNA processing is also illustrated in figure 3.27. Figure 3.27 at the bottom. Okay, so look very carefully at figure 3.27, and towards the bottom half, do you see a light green, well, a green blob, a green ellipse with the word SNRNP? And to the left, you see a bracket. It says exon spliced together. And to the right of the green blob, you see another bracket that says intron snipped and deleted. This is what I'm talking about at this step of the central dogma pathway. RNA processing is also hinted at in figure 3.27 in your textbook. The name of the machinery that does the RNA processing is either SNRNP, as your textbook is telling you. Okay, SNRNPs are called SNRPs. If you look at your textbook, it does tell you that. SNRNP stands for small nuclear ribonucleoprotein. That's a mouthful, right? Small nuclear ribonucleoprotein. That's a SNRP. The video also labeled it by an alternative phrase. The video said spliceosome. Okay, so these are synonymous terms. A SNRP and spliceosome are basically the same thing. Okay, so we're going to tell you about what these processes involved in more detail in about 3 minutes. Okay, so hold on if you're still, you know, we're taking a lot of complex things and we're trying to get you into the game here. All right, so RNA processing takes place by the machinery of the SNRP. That builds the mature messenger RNA you see in the central dogma pathway on your screen. That mature messenger RNA is then going to be exported from the nucleus, right? Because everything we've said so far is taking place in the nucleus of the cell. The mature messenger RNA is exported from the nucleus through the nuclear pores. If you don't remember what the nuclear pores are, take a look at, you could look at figure 327. Probably you might get a better image on figure 324, 3.24. Look for nuclear pore. So the mature messenger RNA is threaded through the opening of the membrane of the nucleus, which is called a nuclear pore. Okay. Once that mature messenger RNA is outside the nucleus, ribosomes are going to manufacture the polypeptide, and that's through a process called translation. Okay, so translation. And the machinery that does that is the ribosome. Okay. Now, the whole process of translation itself is really involved. In fact, I mean, take a look at figure 3.29. Figure 3.29, right? Your textbook devotes an entire page of cartoon diagrams to the process. And I want to ask you to spend your private time reading through that and thinking about the different steps, okay? Well, we're going to get you up to speed on what this is supposed to mean. So the mature messenger RNA leaves the nucleus and is then translated by the ribosome, okay? The ribosome manufactures a polypeptide. Now, if you've had any biology in the past, a lot of times students think polypeptides and proteins are basically the same thing. And they are, but there is an important distinction, a very important distinction. And that distinction is when a newly manufactured polypeptide is made, that polypeptide is not physiologically active yet, in large part because it is unstructured. It is unfolded. It does not have a correct geometry yet to do its job. Okay. So the polypeptide is then going to have to be folded. Okay, so let me give you an analogy. If the polypeptide were a shoestring, let's say you have a shoestring that you're holding between the fingers of your two hands, and you just have this shoestring and it's loose and it's kind of limp and it's all wiggly. It's like a piece of wet pasta, a noodle, right? It's just limp and floating around. There's no structure to it. If we were to take the shoestring and tie it into a bow, like you do when you tie your shoes, then we take the shoestring and we give it a defined shape. In the analogy, you give it the shape of a bow. Okay. When you fold the polypeptide into a specific defined shape, it then gets closer to its true biological activity. Okay, so the folding of the polypeptide will take place to build the protein. The folding is sometimes helped along by a special category of proteins called chaperones. Okay, chaperone proteins assist the folding process. It's kind of like having a chaperone when you were in grade school help you along on a field trip. They make sure the field trip goes smoothly, right? So the chaperone proteins help the polypeptide fold into a protein. Now, some proteins can be physiologically active once they're folded. Some of them can, some of them are. Other proteins might go through some additional processes, which your book doesn't really make a big deal at, but as someone who is a geneticist, I want to tell you about this. Some proteins go through additional things where we have post-translational modifications. And by that, I mean additional small groups of atoms like methyl groups, CH3 groups, things like this can be added or removed from the protein. And that tweaks the structure of the protein just right to have the absolute perfect shape to the protein that now we truly get a physiologically active protein. That's involved, right? This is complicated. We could take any major step in this pathway and give you a 2-hour lecture on it without any problem. Okay, so let's see if we can summarize what's going on here and make sure you appreciate some things. First off, I want to highlight that everything I'm currently outlining in green everything being outlined in green is taking place inside the nucleus of the cell. It's happening in the nucleus, okay? That's its physical location. All right. Everything that's outside the green, and I'll just be very over the top about this, everything that I'm going to highlight in orange is actually taking place in some area of the cytoplasm. Okay, so we have exported from the nucleus. Right, this is happening in the cytoplasm of the cell. Okay, folks, before I go any further, would you like to ask a question about any of this? Okay, so let's see if we can take this detailed version of the central dogma and perhaps talk about some of these major processes like transcription, RNA processing, translation. We need to talk about these in a bit more detail so you get the basic idea. Okay. Who knew that all this complexity was happening inside your cells all the time simply to allow us to say we are alive, right? We take it for granted, but a tremendous amount of invisible, complex processes are happening inside our bodies at this moment. Okay. Exploring the various parts of the central dogma pathway. All right, first off, let's take transcription, right? Transcription is that first part. Transcription. This is the process of taking the instructions in the DNA and building a piece of RNA from it. This is the process of decoding information in the DNA and making RNA from those instructions. Okay. Now, as we said, the machine that does the transcription is RNA polymerase. You know, as you get into higher level courses, like if you were to take my genetics course, I would tell you that there's not just one version of RNA polymerase. There are several different versions of RNA polymerase that do different things. But for right now, we're just going to tell you there's RNA polymerase, okay? So the machine that does the transcription is RNA polymerase, okay? And when RNA polymerase does its job, when transcription is taking place, there's basically 3 major types of RNA that can get manufactured, okay? There's actually more than that, but we're starting at a more simplistic level, okay? There are 3 major types of RNA that get made from the process of transcription. Here's what they are. We get messenger RNA. Okay, this is also known as mRNA. Okay, this is what was referenced up here in your flow chart. Immature messenger RNA, mature messenger RNA. That's what I'm talking about at this moment. Okay, so messenger RNA, what is it? Why does the cell make it? Well, the messenger RNA is ultimately going to be the template for the ribosome. It's going to be the template for the ribosome to make the polypeptide. This will ultimately be used by ribosomes to make polypeptides. Okay. A second type of RNA is known as transfer RNA, tRNA for short. And you might say, well, what the heck is that? Well, I would, let me ask you this. Let me ask you this in the chat window, here, okay? In figure 3.29, what color is the tRNA? What color is it? What color is the transfer RNA in figure 3.29? Come on. What is it? What color is the tRNA? Okay. Take a look at figure 3.29. In the upper right corner, you're going to see it. Okay, good job, folks. You got it. Good job. Absolutely, right? It's a light blue color. Okay, so now you know what we're referring to as the tRNA. So when you look at figure 3.29, you can say, okay, that's the transfer RNA. Good job. All right. So the transfer RNA, what is its purpose? The purpose of the transfer RNA is to bring amino acids to the ribosome, because remember, when you make a polypeptide, you have to link amino acids together with peptide bonds. Okay, so the transfer RNA, think of the transfer RNA as equivalent to the Federal Express delivery guy. The Federal Express delivery guy that delivers amino acids to the ribosome. This molecule's function is to deliver amino acids to ribosomes. That's its job. Okay. A third type of RNA is called ribosomal RNA, or rRNA for short. Okay. Ribosomal RNA is significant for two reasons. Number one, if you think about the machine that we call the ribosome, a curious person might ask, well, what is a ribosome made of? And the quick answer is that a ribosome is made of proteins and RNA, which is called ribosomal RNA. So I'm telling you that the ribosomal RNA is actually a physical structural piece of the ribosome. It makes the ribosome up, okay? This is a structural component of the ribosome. And another important piece of information we share with beginning students is that the ribosomal RNA is actually the piece of the ribosome that's responsible for creating the peptide bonds. And is responsible for forming peptide bonds between amino acids. Okay, so folks, these are the three major types of RNA that get made from the process of transcription. The enzyme that makes these different types of RNA is called RNA polymerase. Do you have questions right now? All right, so we're still in the topic of transcription here, okay? And I want to give you the basic idea of transcription. Transcription, you take a DNA template and you make RNA from it. Okay. The basic rules of transcription are as follows. Okay. If you have a fragment of DNA and it's going to be used to make a fragment of RNA, okay, here's what you're going to observe. Okay. If in the DNA, I have a specific kind of nucleotide, which is named for the nitrogen base of the nucleotide called adenine. If I have adenine in the DNA, then what you're going to find is that I will end up with uracil in the RNA. If you end up with thymine in the DNA, then you're going to end up with adenine in the RNA. If you have cytosine in the DNA, you're going to end up with guanine in the RNA. And if you start out with guanine in the DNA, you're going to end up with cytosine in the RNA. Okay. Now, if you don't know what I'm talking about here, if this is all Greek to you, okay, it's not Greek, but it might seem like it's Greek. Okay, first off, take a look at either figure 3.27 or figure 3.29. On the lower right, in the lower left corner, do you see something that's the key? The key. And do you see adenine, guanine, thymine, and cytosine for these different pictures here? Okay, this is what I'm talking about. Okay, so maybe we should do this. Remember that nucleotides are named for their nitrogen bases. And if you don't know what the nucleotide is, you got to go back to chapter 2 and look that up. Nucleotides are named for their nitrogen bases. And so basically we have these nitrogen bases, adenine, thymine, cytosine, guanine, and uracil. Okay, these are just a list of the nitrogen bases. Okay, so let's see if you can do an example here. Okay. Now, typically, rather than writing out the full name of the nitrogen base, we'll abbreviate it with an appropriate letter, right? So adenine will become A, uracil will become U, thymine will become T. Okay, we'll do it like that. So it's a little bit easier to write down and make sense of it while we're writing things down. Cytosine will become C. Guanine will become G. Okay. All right, so folks, let's do this. If I give you a snippet of DNA with the following sequence, okay, can you give me the RNA that would correspond to it in the chat window? What would be the RNA that would be made from the DNA? What would be the RNA that could be made from the DNA? See if you can put that into the chat window. What would be the RNA that's made from this DNA? Okay. So Noah has an answer in the chat window. How about the rest of you? Okay, thank you, Rachel. Let's get one more person. Okay, Karen, very good, very good. Looks like you guys got it right. So just for everybody's benefit, we'll go slowly here, right? If the first letter in DNA is G, then the corresponding letter in RNA is going to be C. If the letter in DNA is C, then the corresponding letter will be G in the RNA. If the next two letters of the DNA are G, then we get two C's. If the next two letters in the DNA is T's, then we get two A's. If the next two letters in the DNA is A, then we get two U's. And then we'll just finish this out. C, G, A. There we go, folks. Okay, so let me ask you in the chat window. Are you with me? Is this making sense? Great. All right, I think there was a question up here. So Rachel is asking, is this a set for DNA and RNA, or are there more and there's only 4 for the example? Okay. All right. So Rachel, let me give a little bit more background here to answer your question. Okay. There are 4 nucleotides for DNA. Nucleotides are named for their nitrogenous bases. So the DNA nucleotides are adenine, thymine, cytosine, and guanine. There are 4 nucleotides for RNA. They are adenine, uracil, cytosine, and guanine. So Rachel, these are the specific nucleotides for DNA and the specific nucleotides for RNA. Are there others? Truth be told, there can be some others, but 99.9% of the time that we are worried about with biology, these are it. Okay, so we're giving you the examples that are 99.9% of the time correct. Okay. Any other questions? Okay, so in our example here, which you all did beautifully on, the DNA sequence, which I'm highlighting right now, would be used as a template by the RNA polymerase enzyme to make the snippet of RNA that you all gave me in red. Okay, that's transcription. Okay, next. We need to go to RNA processing. Okay, so we did transcription. Next is RNA processing. For RNA processing, you know, what exactly is going to happen here? Well, it's complicated, but it's understandable. I mean, we can do this. Okay, and so let me get my RNA processing. Okay. Let me just get another line of separation here. Okay, so RNA processing, RNA processing is the process that occurs when you take the immature RNA and you manipulate it to form mature RNA. In the process, sections of RNA that are known as introns get cut out, and sections of RNA that are known as exons get glued together. Okay. In this process, machinery of the cell, remove introns and glue exons together so that immature RNA, immature messenger RNA, can be converted into mature messenger RNA. Okay. The machine that does this, once again, the machine that does RNA processing is called the SNRP, which, you know, the SNRP, which is known as SNRNP or spliceosome. These are equivalent terms, okay? So there's a comment from Kelly in the chat window. Do we have lecture first, then the exam on Wednesday? Yes, Kelly, you have normal lecture on Wednesday. After the normal lecture Wednesday, you then go into class marker for your lab exam. Okay. All right, so here's what I'm going to do is give you the basic idea for RNA processing. Okay, and to do this, I'm just going to make a little cartoon. All right. So when the cell makes its immature RNA, it's going to be one physical continuous chain of RNA nucleotides. It's going to be one continuous physical chain-like molecule. Okay. But there are going to be defined sections that the cell can identify. So let me do it this way. So right now, I'm simply taking my snippets of green and linking them with snippets of red. Okay. And what I'm trying to show you with this is that the first section of green would be potentially exon 1. The second snippet of green would be exon 2. Third snippet would be exon 3. The first snippet of red could be intron 1. Second snippet would be intron 2. And this whole thing would be immature messenger RNA. In some books, you'll see it referred to as pre-messenger RNA. Okay. And so what's going to happen here? Okay. Let me see here. Oop, don't like that. Okay, let me, okay, well, all right. I don't have a lot of space here, but I'm going to just draw this out. Okay, I want to try to draw it out. Okay, so RNA processing will take place by the machinery of the spliceosome, and you're going to end up with this. You're going to end up with exon 1 glued to exon 2, glued to exon 3. And I'm simply making a vertical line so you can see where they would be, where one exon would be end and another would begin, okay? But there is no physical line in the real cell, okay? And so you're going to end up with this, right? You're going to end up with exon 1, exon 2, exon 3. Okay. The introns are removed. Okay. The introns are removed. And this would be the mature messenger RNA. Okay. Now, okay, I need to stop drawing and get to the next page here. Okay. Let's see. Okay, that's not going to be helpful. Okay, so I'm wrestling with my technology here, which is never good. Let's do that. Oh dear, what happened to my drawing? Where did it go? Okay, my drawing just evaporated on me. Where did it go? Okay, well, computer's doing some wonky things on me. So for the moment, I'm just going to pretend like everything's okay and not mess with it. All right. Okay. So let me do this and make a couple comments here. Okay. In the drawing, the introns are physically removed by the machinery of the spliceosome, and the exons are glued together. Okay. When I was an undergraduate, I was taught that the introns are simply junk. They're discarded and thrown out by the cell. They basically have no purpose. They're kind of evolutionary waste that is meaningless. And I can tell you in the several decades since I've been an undergraduate, that science has discovered that that is really false. Introns are very important. They're not actually used by the ribosome to make the protein, but introns contain control and regulatory information where the introns go off and interact with other machinery of the cell to control when genes are turned on and off. Okay, so introns are removed from the immature messenger RNA and ultimately help regulate the expression of other genes. In other words, introns play a role in turning genes on and off. Okay. All right, folks, do you have any questions right now? Okay, so we've covered transcription, we've covered RNA processing a bit. Okay, the next major thing that I'm going to talk about is translation. We need to talk about translation a little bit. Okay, so that would be letter C here in our notes. Translation. Okay, translation is the topic of figure 3.29. I'm not going to go through every step of that with you in class. I want you to read it and think about what's being explained, but I want to help you get the big idea here in just a minute or two that we have left. Translation is the process of using the mature messenger RNA template to manufacture a polypeptide. And for right now, I'll put in parentheses protein, okay, even though they're not quite the same thing. To manufacture a polypeptide or protein, okay? And so the machine that does this, as you know, is the ribosome. And remember, there's from our conversations at the beginning of chapter 3, our very first lecture in chapter 3, you know that ribosomes are found typically attached with the rough endoplasmic reticulum membrane, as well as sometimes ribosomes can simply be floating in the cytoplasm like a beach ball bobbing in the water of your swimming pool, okay? So the machine that performs translation is the ribosome, and the basic idea is this, okay? When the ribosome is sitting on top of the messenger RNA, as you see it in figure 329, it's going to interpret groups of 3 nucleotides at once. It's going to interpret every 3 nucleotides as a group together at one time in the messenger RNA template. So the basic idea is this: each 3 nucleotides in the messenger RNA is called a codon. Each 3 nucleotides in the messenger RNA is called a codon. Okay. And each codon specifies 1 amino acid. Each codon specifies 1 amino acid, okay? So let me ask you this in the chat window. Let me ask you this in the chat window. How many amino acids would a ribosome stick together if there were a stretch of 15 nucleotides in the messenger RNA? Okay. How many amino acids would a ribosome stick together if there were a stretch of 15 nucleotides in the messenger RNA? Okay, thanks, Noah. How about the rest of you? What do you think? Every 3 nucleotides specifies 1 codon. 1 codon specifies 1 amino acid. Okay, Madison, Rachel, good job. Yeah, the answer is 5, right? How did you get 5? Well, if there's 15 nucleotides, which are interpreted in groups of 3, that means 15 divided by 3 equals 5. So 5 amino acids would be linked together. Okay. So let me give you an example here. Okay. Come on, software. Okay. Oh. Okay. The example is this that I want to give you. If you have the following stretch of nucleotides in the messenger RNA. Okay, so if we had the messenger RNA, if the messenger RNA had this. All right, how are we going to decode this? Well, folks, remember in the video, it showed you the genetic code table. All right, this beast right here, okay? This beast right here, let me bring this up in a new tab. Come on, computer, work for me, please. All right, this beastie right here, okay? Now, I don't expect you to memorize this. I do not expect you to memorize it, and it's not even published in your textbook, okay? So if I want you to use this, I would actually print it on your exam and provide it to you. But okay, let's say CAC, CAC, so I need to find that, CAC, that's going to be histidine. So the amino acid that's going to go into the polypeptide or protein, if you will, is going to be histidine here. Okay, the next one I put out is GAC. Let's look for GAC. Okay, here's G, here's A, here's C, GAC. That's ASP, which is short for aspartic acid. So next we're going to have is aspartic acid. Okay, I'm running out of space here. Next is GGG. So GGG down here specifies the amino acid called GLY, which is short for glycine. Okay, so we're going to put glycine in there. And next we have GGC. I don't know, that might also be glycine. Let me see here. Yeah, GGC is also glycine. Okay, so in our simplistic example in the notes, these are the things that you would get in your new protein. Okay. right. Each codon specifies one amino acid. This information can be found in a published genetic code table. Okay, that's published genetic code table. All right. All right, folks. Questions right now? I know this is a tremendous amount of material. You will have to spend some significant study time on this. Okay. All right. So I don't have a lot of time left. And you know, there is content in chapter 3 I'm not going to have time to cover with you. Okay. I mean, I could spend 6 lectures in this chapter. I've only given you 3. Okay. So I want to highlight cell reproduction a little bit at the very end of chapter 3. Right. All right, cell reproduction. All I can do is make a few statements, folks, for you and point you in the right direction during your private study time. Okay, you know from information in your book that there's basically, so right now I'm on pages 96, 97, 98, 99. You know, I'm going to be referencing some figures here, figure 3.32, for example. Okay, so cell reproduction in animal cells. Okay, like the cells of our body, there are 2 major methods of cell reproduction. These 2 methods are mitosis and meiosis. Okay, let me highlight that, mitosis and meiosis. You probably have come across these words before. Okay, so mitosis. Okay, let's just make a couple comments about mitosis. The purpose of mitosis is to create genetically identical cells. That is its purpose. When a cell has to reproduce and it uses mitosis, the goal is to create a genetically identical cell, a clone of the original cell. Purpose is to create a perfect genetic copy of the original cell. Okay, so mitosis is going to be used by the body, by your body. Mitosis is going to be used anytime you have to heal the body in some way where there's been a wound. For example, you're running outside, you fall down on the concrete, you scrape your elbow, scrape your knee, you get a brush burn. You scrape the skin open, it bleeds a little bit. Obviously, you go in, you clean it off with some soap and water, put a Band-Aid on it, and in the next couple of days, it will heal. Well, how does it heal? It heals because new cells are manufactured through the process of mitosis. So mitosis is used to heal the body, and it is also used to grow the body. Obviously, you are a little bit taller today than you were when you were 2 years old. You grew up. Well, how did that happen? One way it happened was making more cells to get you bigger. Okay, so mitosis will also be used to grow the body. Okay, so that's mitosis in a very quick nutshell. Meiosis, well, meiosis, so let's put it this way. Mitosis will be performed by all cells of the body except your sex cells. Okay, meiosis will be performed by the cells of your body that manufacture sex cells. Okay, so meiosis is going to be performed by the stem cells of your body that make your sperm and egg. And so I know I'm out of time. Let me just make two statements and I promise I'll let you go. The first statement is the purpose of meiosis is twofold. Twofold from the perspective of the first thing that has to happen is we have to cut the chromosome number in half. We have to cut the chromosome number in half. Okay. Meiosis seeks to reduce the chromosome number by half. And so you remember that means typically human cells have 46 total chromosomes, but a sperm or egg cell is going to have 23. So we go from 46 to 23 chromosomes. And the second thing that meiosis seeks to do is to take the DNA of the chromosomes and swap it around, kind of like you're shuffling a deck of cards where you take different physical pieces of chromosomes and you swap them around. And the purpose of that is to intentionally produce genetic variation in the cell's DNA so that when sperm and egg meet, the resulting child is not a clone of you. Okay? To swaps pieces of DNA intentionally to create genetic variation. This way, our children are not clones of us. Okay. All right. So, there's plenty for you to read about these major topics at the end of chapter 3. There's plenty of you to read and study in chapter 3. Okay. I'm going to stop right there, folks. This completes my coverage of chapter 3. I could spend more weeks on this, but there's just not enough time to complete all things in the curriculum, which is my way of saying there are topics you must study on your own. Okay, that's the only way it's going to happen. So, do you have any questions? Okay, let me remind you, later today, I will place your first lecture exam study guide in Wiley Plus. And don't forget, the most important thing that you're doing right now is preparing for your lab exam on Wednesday. That's the first major exam of the semester, and you want to get off to a good start, okay?